Hello, everybody. All right, so you're watching this video because we're going to go over the Unit 7 study guide. This is the unit that we did together in class, took notes together in class. You should have the study guide glued into your paper notebook. Hopefully you took those home and you're good to go. And we're going to go over the answers right now so that you feel confident when you're walking into your unit exam later this week. A lot of people are asking like, oh, well, do I need to take this exam? It's the exam. It's going to be a way to improve your grade. I know a lot of people aren't happy with their grades right now, and we always talk about in class that exams are your #1 way to bring your grade up. So if you're like, I need to improve my grade, doing well on this will work. Good news for you, there are no short answer questions. It's all multiple choice. So this is even easier. Then you add on top of that, I'm expecting that you're going to have your notes out. I'm expecting that you're going to have like a study guide or something like that there to help you. So this should be pretty much a walk in the park. You'd be kind of missing out on an opportunity if you don't take advantage of this. So I would definitely recommend that you take the quiz at the end of the week. Have your notebook out, have your study guide out, have your concept map out, have all that stuff out, and you should be able to rock and roll this thing. The only thing, and I will mention this on the other video that talks about how to take an online test through Jupyter, is once you click on start the exam, you got 2 hours to finish that exam. Okay, and there's reasons I'm doing that, but basically don't click it to start unless you're ready to sit down and be there for a good chunk of time. Now the questions, there's like 33 multiple choice questions, no short answer at all. And so all you need to do is run through those 33, give the correct answer. It should give you your score at the end, and there you go. That's your test. So be ready. Now it's not gonna take you 2 hours, but be ready to take a chunk of time in case you need it. Okay, all right, so let's jump into going over the information that's on that test, help you out. So I've got the 3 papers that we glued in. First one looked like this. Second one looked like that. And the third one looked like this. Now the third page was a short answer page. I'm gonna go over the answers on the short answer at the end, but these are turned into multiple choice questions now. They are not short answer questions any longer. Okay, so let's start up at the front. Okay, let's get everything set up here so you can see this stuff. Lock it down. Here we go. All right. Draw an example of a DNA nucleotide. Draw an example. There's a typo for you. Draw an example of an RNA nucleotide. How are they different? Okay. When we did in class, DNA nucleotide, very basic. Got this P with a circle. You got this pentagon connected to a rectangle we call the base. Okay, hopefully you remember that this thing here is called a phosphate. This thing here is a sugar. And this is the base, as I've labeled it right there. Draw an example of an RNA nucleotide. Haha, tricky part, because look, there's a phosphate. There's a sugar. There's a base. So you're saying, what? They're the same? Yes, the nucleotide is the same, except the parts are a little bit different. Do you remember what the parts are? Do you remember how the parts are different? Think about it, think about it, think about it. The sugar in DNA is deoxyribose. The sugar in the RNA is ribose. Still a sugar, just different sugars. And what about the base? Hopefully that's the one you remember, because you're gonna need to know that. Remember the bases, remember the letters, A, G, C, and T. So what are they in RNA? Click, click, click. They are A, G, C, but no T. Remember, instead of a T, there is a U. That is correct. So that explains how they are different. Okay? Both of these are nucleotide, nucleotide, nucleotide. These with this sugar make DNA. These with this sugar make RNA. All right. Number 2, explain how DNA, RNA, and proteins are polymers. Okay. So that's that word, polymer. Polymer, if you remember in class, is just is a repeating thing over and over and over again. That repeating thing in DNA are these nucleotides. So it goes nucleotide, 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 nucleotide. In RNA, that repeating thing is a nucleotide, 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 nucleotide. That repeating thing in a protein, though, is an amino acid. Amino acid, amino acid, amino acid, amino acid, amino acid. So it's still a string of things put together. Those things are called monomers, okay? The monomers for DNA and RNA are called nucleotides. The monomers for a protein, the thing that repeats over and over again, is amino acid. So people get tripped up when they see these words. They're like, wait, what? Just remember, poly means many. So when you put many of these together, you make a polymer. Mono means one. So each one of these is a nucleotide. Put a bunch of nucleotides together, you make DNA and RNA. One amino acid is one of these. Put a bunch of amino acids together, which is a polymer, it makes a protein. Okay. These are very generic terms. These are very specific. All right, number 3. What does antiparallel mean? Antiparallel basically means that they are parallel lines going in the opposite direction. Parallel in opposite direction. Why is DNA considered to be like that? Well, remember it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, and then keeps going that way. But on the other side, it's flipped upside down. I don't know if you guys remember that. So you got sugar, practicing my up-down skills here, phosphate, sugar, phosphate. Hey, if you think it's easy drawing a pentagon upside down, you just try it. You just give that a shot. Okay? So on this side, they go this way. On this side, they go that way. That's why DNA is considered anti-parallel, because they're parallel lines. They're just going in opposite directions. All right, number 4. Please do not miss this on the test. Oh my gosh. What are the 4 bases for DNA? If you want to get super fancy, it's adenosine, guanine, cytosine. Oh, I think I misspelled that. Cytosine and thymine. If you can just remember A, G, C, and T, we're probably going to be good. Okay? But just, I don't want you to think we're just doing letters from the alphabet here. They actually stand for different bases. All right, number 5. Which part of the molecule makes the entire molecule negative? Okay, you had a gel electrophoresis lab on this. Remember how the gel made the electricity would make the DNA move? Why did the DNA move? Because it was negative. What makes it negative? Those little phosphates. Oh, those little phosphates, the P's with the circles. They are negatively charged. So if you look at this picture I drew up here, you got negative, 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 negative. And that's just a little piece of DNA. Imagine like this thing hundreds long, how many negatives there'd be. It's super, super negative molecule. So that's what makes it negative. The phosphates have a negative charge. Number 6. If one strand of DNA has the sequence G, T, C, T, A, C, A, T, G, what would corresponding DNA strand be? Okay, tricky thing on these is make sure you look at it and go, I'm going from DNA to DNA, not RNA, not mRNA. It's DNA to DNA. So that means I'm going to use these. And you should know which ones pair with which ones. Oh, we missed that question. A pairs with T, G pairs with C. Okay, so if I see a G, C. If I see a T, A. I see a C, G. I see a T, A. I see an A, T, C, G, A, T, T, A, G, C. There you go. That's how it works. You just have to know what pairs with what. tricky part, DNA to DNA, because there's going to be another question that says DNA to RNA pretty soon. What is a DNA fingerprint? Okay, very simply, it's that. But you got to explain more than just that. You got to explain what it is. DNA fingerprint, it is a pattern of DNA pieces unique to an individual. If I take your DNA, chop it up with an enzyme, and I take somebody else's DNA and chop it up with the same enzyme, it will give me different pieces with different lengths, different sizes. And so when I run it on a gel, your pattern is going to be different than somebody else's pattern. And that's why 4 different people, like you see here, will have different bands or different patterns, because each of these are pieces of DNA and they have different size pieces. And we'll get into why they different size pieces in just a moment. Okay. Look at the example gel from gel electrophoresis lab. Which suspect left DNA at the crime scene? And how do you know? All right, so blood stain is our crime scene. So we took the DNA from the crime scene, we ran it on a gel. It gives us this pattern of bands. So you look and you say, all right, who has the same pattern of bands? All right, Bob and Sue and John and Lisa, they all have the top one, so that doesn't help. All right, second one. Bob has the same, John has the same, Lisa has the same. So Sue's already out. Next one, just John and Sue, but Sue was already out from the first one. So, I mean, I don't even have to go any further. Boom, there it is, John. It's John. And how do I know that? His DNA fingerprint, or you could say DNA pattern, matches the pattern from the crime scene. And we talked about in this class, that just tells you that he left DNA at the crime scene. That doesn't tell you what he did when he was there. Nah, it looks a little fishy when, you know, there's like, let's say, I don't know, like there's been an attempted murder or robbery or something, and there's a glove, and the glove has blood on it, and the blood is your DNA. Yeah, good luck trying to explain that one. All right, number 9. Describe the central dogma of biology. Easy. All the information starts in DNA, and we take the DNA and we take the information into mRNA or just RNA, and then that helps us to make a protein. Okay, it stays in your DNA. We'll talk about why you have to make this RNA. Why can't you just go DNA to protein? Why you gotta have this thing in the middle? It's because your DNA can't leave the nucleus. Your DNA has to stay in there. It keeps it protected. Plus, it's like super huge, and it's like trying to get like a string out of your house when the ball of string is like the size of your kitchen or something like that. It's just not going to happen. All right, there's 2 processes here. This process of DNA mRNA is, hopefully you got it right, transcription. And the process from mRNA to protein, going to 2 different languages. That's the way to remember it. From the language of NA to the language of protein, translation. DNA to RNA is called transcription. RNA to protein is called translation. What does DNA do? This is always the funny thing, because everybody thinks, oh, I hear it on all the crime scenes. It must do some really important stuff. It doesn't do anything. What it does, I guess you could say, is holds information on how to make all the proteins in your body. And the proteins do the work. That's key feature. The proteins make you have 5 or 6 fingers. Proteins make you have blue, green, or brown eyes. The proteins make sure you have a certain hair color. You know, whether you have hair on your knuckles or you don't have hair on your knuckles, that's decided by proteins. So you think, well, then what is the point of DNA? DNA just has the information. It's the blueprints of how to make all the proteins to do all the work. That's how you have to think of it. Maybe put that in parentheses. Blueprints. All right, number 11. In transcription, where does the information start at? What is created at the end? Okay, so we go back to our central dogma. This is key. Transcription. Where is the information at? It's in DNA. What do we have at the end? mRNA. Information starts in the DNA. At the end, info is in mRNA. There you go. Straight off your genetic central dogma of biology. All right, that's page 1. Page 2. Dun, 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 dun. All right, number 12. Explain the differences between DNA and RNA. Okay, so we got a couple things. All right, let's see, how should we do this? Let's do a nice little T-chart. Put DNA over here, RNA over here. Okay, first thing that should jump out at you is there's a different letter. Okay, why? Because the first letter is the sugar. This is deoxyribose, this is ribose. So this has deoxyribose sugar, and this just has ribose sugar. So the difference in letters is the difference in sugars. Okay, what else? DNA, if you ever draw it, kind of looks like that. You got the little da 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 da. Okay, RNA, woo woo woo, but only one side. So what's the difference there? This has got single strand, this has 2 strands. This is just a line, this is a double helix. So there's your differences right there. Because this has 2 strands holding together like a nice twisted ladder, this is very strong. This is very weak. Your RNA only lasts for hours, maybe up to a day at the most. It is very weak molecule, falls apart, which is good because it's like sends you a message and you're done with it. You don't need it anymore. Break it apart, make a new message later on. Okay, so single-stranded, double-stranded, sugars are different, and this is very strong, this is very weak. And then the most key important one here are the bases. We mentioned that already. A, G, C, and T. I'm going to make that stand out. And then over here, A, G, C, same 3, except there's no T. There's U. Which, do you remember the name? Do. That's right, uracil. Okay, so it has uracil, not thymine. All right, that's good differences there. Explain the role of RNA polymerase. Okay, so RNA polymerase is this molecule, comes in. It unzips the DNA like a zipper, just zip, because your DNA kind of looks like a zipper, if you want to think of it like that, and it has the other side, and the RNA polymerase comes in right here. This is your RNA polymerase. You might be thinking, why is it called polymerase? Because it makes a polymer of DNA, of RNA. It makes the polymer of RNA. Okay, so as it opens it up, it's also doing another thing. Let's see. Okay, it's reading the DNA. And so if it said, oh, this is an A, it puts a U. If it's a G, it puts a C. If it's a C, it puts a G. If it was a T, it puts an A. And if it's an A, right, there is no T, there's only U, so it puts a U. So it makes the mRNA. So what does the RNA polymerase do? It unzips it, and then it makes the RNA that goes with the DNA. So if this, it just matches them up, but it makes RNA, not DNA. It makes RNA. Very key point right there. Okay, that's in transcription. Keep those same, because where are we going from? DNA to RNA. All right, number 14. Where does transcription take place in a cell? Where does translation take place in a cell? Okay, if you remember, we said a cell, and then that little area right there is called the nucleus. And the area outside the nucleus, but still inside the cell, is the cytoplasm. Okay, so inside the nucleus is where the DNA is at. So it's in here. It's not. It's like its own little special room, safety storage compartment, safe, however you want to think of it. DNA is all in here. Okay, And so the DNA can't leave. So it makes complete sense that if you're going from DNA to RNA in transcription, you have to go where the DNA is. So the transcription takes place in the nucleus. Well, once it makes the RNA in the nucleus, the RNA comes out of the nucleus into this little area here, which is called the cytoplasm, to do translation. So that is in the cytoplasm. Okay. Number 15. Why does the information on DNA need to be coded onto mRNA and then used to make a protein? Why doesn't the ribosome and the tRNA just read the DNA directly? Okay, so this goes back to our little diagram here. The DNA is inside of here, and it's very well protected. And so you want to keep your DNA here, and all the ribosomes and the tRNAs are all out here in the cytoplasm. And so if you let that stuff have access to your DNA, it would possibly damage your DNA. And when you damage your DNA, that's when bad things happen. That's when you make nasty proteins that don't do what they're supposed to do, or you make proteins that do worse things than not working and cause problems in your body. Cancer is one of the many things that goes wrong when proteins are made incorrectly. So you don't want to screw up your blueprints. You screw up your blueprints, you're going to have screwed up cells. Okay, so you protect your DNA. So you have to keep the DNA protected. Have to keep the DNA protected. Protected in the nucleus. And just like a king or queen sitting in a castle, you know, they're going to stay protected behind the walls of the castle. If they got to send a message out, they're not going to get on a horse themselves and walk out the castle. No, they're going to get some messenger to do it for them, right? So what does DNA do? DNA makes, so the DNA codes for RNA, for a messenger RNA to send the code out. He's like, you go send the message. That way, if they kill you when you get message get there, it didn't hurt me. So the mRNA goes out to where all the machinery is at in the cytoplasm and deals with that harsh environment out there, whereas the DNA gets to stay nice and safe inside the nucleus. There you go. That's why you have to make a messenger first. You have to make the messenger to send out the message to where everything else is at. All right, number 16. In translation, where does the information start? Where is it created? Okay, so this goes back to that central dogma we had right here. Where does the information start in translation? You've already made the mRNA, so that's where it starts. So it's starting with the mRNA. And you're going to make it into a protein. And what a protein is, as we already mentioned, is a chain of amino acids. Okay. What is the role of tRNA during translation? Okay, tRNA, that's our little clover friend. And he has on the top something that he brings. That is the amino acid. And on the bottom, he's got three little letters. Oh, let's just say A, G, G. And that's the part that's going to match up with the mRNA. So that is called the anticodon. Okay, so this is the anticodon, matches with the codon of the mRNA, and it has the amino acid. So what is his role? His role is to bring, I'm going to abbreviate amino acid AA, bring amino acid to the ribosome that matches the codon. Okay, what's the role of the ribosome? Ribosome, so if this is my mRNA, got my ribosome right here. Okay, so I've got one little codon right here, got another little codon right here. I've got a tRNA inside of it, dropping off its amino acid. This guy now has already dropped off his amino acid, so he's empty, and the amino acids are all chained together going out the top. Okay, so there's my tRNA, anticodon matching with codon. So where is the ribosome doing? Oh, the ribosome is this circle right here. Ribosome. He just creates a nice little house for all this to happen. It's like creating a workshop. He's got a nice little place for the tRNAs and the mRNAs to match themselves up nice and safe. So what is his role? To create a place for tRNAs and mRNAs to, word that jumps to mind is hook up. Probably not exactly right. To connect, let's just keep it really platonic like that. They just got connected, that's all. All right, number 19. So let's get into like more of a bigger picture here. Your fingernails are made from a protein called keratin. In order to make a longer fingernail, your cells in your finger need to make keratin nonstop. That's why they just keep growing and growing and growing. What steps would have to take place from the moment your brain signals the cells to make keratin? No, use basic steps. Okay, so your brain sends a signal to your finger, says, oh, fingernails, need more fingernail. So that then goes to the DNA. So the signal goes to the nucleus of finger cells. The DNA code, the DNA gene for keratin gets turned on. What does that mean? It codes for an mRNA. mRNA leaves to the cytoplasm. Again, these are very basic steps. 4, the mRNA codes for a protein. The keratin protein, and then 5, the keratin is added to your current fingernail. And makes it grow a little bit longer every day. Now, as you sat there and watched it, you ain't gonna see anything happen because there's so small of molecules being added to your fingernail that in the time that you would sit there and watch it, you're not gonna see anything happen. But what typically happens is it just goes on and on while we sleep, while we're awake, while we're doing all the things we're doing. And then we look at it like maybe 5 to 7 days later and go, huh, look, my fingernails are a little bit longer. Look at that. It's happening. You just don't see it happening. Okay, number 20, another big picture question. Diabetes type 1, we talked about that. They cannot produce functional insulin, which is a protein. So they can't make a correct protein. They can make a protein, it just doesn't work the way it's supposed to, or work at all. What does this say about their genes? So their genes, which are their DNA, normally would make an mRNA, which then would make the protein. So you have to say, are they still making protein? Yes, still making protein. But the protein is not the right protein. So they still made an mRNA and they still made a protein. So what's different about it compared to regular insulin? There must be something different about the amino acids in the protein. Well, if there's different amino acids, then that means the code in the RNA was different. If the code in the RNA was different, that means the code in the DNA was different. So the code in the DNA for insulin, the blueprints for insulin, must not be correct. Still making protein, it's just not the correct protein. That's why it doesn't do what it's supposed to do. Okay, and we'll talk about protein folding a little bit later. I think we're gonna get into that in about 3 weeks or so. Okay, cool. All right, those are all what were the multiple choice questions. Now let's drop into the short answer questions here. Okay, the gel. Okay, so it asks you to basically describe why running electricity through the gel like does what it does. So let's just go over it. So let's draw a gel. Hopefully this picture looks familiar to you. You had your wells. Okay, where you put the DNA inside. So you put the DNA in there. Fill up the well with DNA. It's all nice and stays right there. And it will stay right there. Unless you do something, it's just going to sit right there in the gel, not go anywhere, not do anything. So the DNA is going to sit there. So what did we do? We put the DNA, we covered it in with buffer, if you remember when we did this in class. And then we did what? After we put the lid on, plugged it in and turned on the electricity. Why? Because when we put on the electricity, it makes one side go positive, 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 positive. Actually, I got that all wrong. Good for you to remember. Remember my mistake. Negative, 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 negative. And on the other side, it's positive, 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 positive. Okay. Some people did put it that way and they had their gel flipped and it went the wrong direction. But you don't want to do that. So you want to make it on the negative side. Why? Because DNA is what charge? Negative. It's negative because of those phosphates. Remember those phosphates have a negative charge? Okay, so DNA is very, 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 very negative. Lots of negative charges. Tons of them. So what happens when you put a negative next to a negative? It goes, get me the hell out of here. So the DNA goes, I'm moving away from the negative. I'm going that way. And what do negatives like about positives? Well, they like to be attracted to them. So they're like, oh, this is doubly great. I'm getting away from my negative. I'm going towards my positive. So the DNA just keeps on moving, keeps on moving, keeps on moving all the way down. Now, if you run your gel too long, that DNA will go all the way down the gel, out the back end, and you'll have nothing here. So you have to be very tricky about running it, but not waiting too long because it'll just run right off the gel. Okay, so that's what the electricity does. Electricity creates the negatives and the positives. The phosphates are already on the negative. The DNA is in fragments or pieces in here. Okay, you got that. So you create a pattern. Get a pattern of DNA. Why do you get a pattern? Why doesn't all the DNA just keep traveling together? Well, because of the fragment size. Some are small, some are little small pieces, and some are big. And so why does that matter? Because going through this gel is like going through a series of caves inside of a mountain. And when you're a very big piece of DNA, you don't move through caves very well. You get all stuck on things and caught on the side, and it takes a while for the big pieces to go through the caves. So they go slow. So up at the top of the gel are where your big pieces are at. Well, the small pieces, they can fly through the caves real easy. So they're like, woohoo, and they move a lot faster than the big pieces. So the small pieces are down at the bottom of the gel. The bigger pieces are up at the top of the gel. And so the size of the fragment definitely makes a difference. Smaller pieces at the bottom, bigger pieces at the top. And what that pattern is, that DNA pattern is called your DNA fingerprint. Great. So that kind of just explains how this all goes together. All right. All right. Next thing. Number 22. This was the one I thought everybody was going to do awesome on and get like full credit for. So we're doing a DNA strand. This is the sequence of DNA. I want you to make the RNA, and then from the RNA, make the protein. So you literally go, T goes with A goes with, that's right, U. There is no T in RNA. C goes with G, T goes with A goes with U, C goes with G, T goes with A, C, G, U, A, U, C, G. Losing space here. What is that? 4 of them in a row. So it goes A, U, A, G. Looks like I should have spaced them out a little bit more. Okay, so that is how you get your mRNA strand. All right, now to go from your mRNA strand to your protein, you're going to need some help. You're going to need a genetic code. So I'm going to pull that up for you right now. Let's see where I put my images at. 1 second here. Images. Pulling it up, pulling it up, pulling it up. Okay, and remember, to go from mRNA to protein, you're going to need to break these into codons. So every 3, AUG, AUG, ACC, GGU, AUA, UUU, CGA, AAA, UAG. Hopefully it breaks itself down like that so it'd be easy for you to find when you got to go to the genetic code. If you remember our genetic code, it is the little circle sheet. So let me pull this up for everybody to see. This is on the actual computer. Right here. All right, so let me screen share this with everybody real quick so you can see it. This guy right here. You guys remember this guy? Okay, where you go start with the first letter in the center, and then you go second letter is the second ring, third letter is the third ring. So our first one is AUG, so I go to A, and then to U, and then to G. And it brings me to methionine. The 3-letter abbreviation is MET. The 1-letter abbreviation is M. We were using the 3-letter abbreviations class, so I'm going to use MET. Okay, so I'm going to use that. I'm going to go back to our picture here. I'm going to use that to guide me. So we said AUG makes MET. The next one is AUG as well, so that makes it nice. AUG is MET again. The third one is ACC. So I go over here, ACC, which is threonine, or the THR. Then I get GGU. GGU is glycine, or GLY. And then I get AUA, which is isoleucine. Then I have UUU, which is phenylalanine. CGA, which is arginine, or ARG. AAA, which is asparagine, ASN. And then hopefully this last one looks a little bit familiar, UAG. Remember, you are gross, that's a stop codon. Remember, you are gross, you are annoying, you go away. You are gross, you are annoying, you go away. Okay, so remember, you run into any one of those 3 and you are hitting stop, and you just stop right where you're at right there. Okay, so that takes care of that. Hopefully you got that one at least decently down so you can work on that one. Let's see. Last question, we need to talk about translation. And I'm going to actually show you the image that you're going to see on your exam. So for all the folks who are taking the time to watch this entire video, kudos to you, because you're going to get special help with exactly what you're going to see on your test. So let's go back to screen share so you can see what I'm talking about. Go away, window. All right, here we go. This guy right here. Blow it up a little bit. Okay, so this is what you're going to see on your test. I'm going to ask you to identify these letters. So you should have seen this. This should be copied into your notes. This information is also available on the internet, as well as this video here. So let's go over it. Let's start with A. A is talking about this whole molecule right here with a little thing at the top and an anticodon right here. Oh, that's a tRNA. Okay, B. What is B? B is pointing to this chef's hat mushroom looking thing that houses all this stuff, creates a nice location. B is ribosome. This is your ribosome right here. All right, C. C is pointing to the little boxes and circles and triangles that are going to be added out the top of the ribosome. So they got 3 letters on them. Hopefully that rings a bell. Those are your amino acids. So this is your amino acid chain. All right, D. D is pointing to the mRNA, but a specific part of it. It's pointing to this part right down here, AUG. So if that's on the mRNA, that must mean it's a codon. So that is the codon on mRNA. The part that matches with the codon off of the tRNA, which F is pointing to, that is your anticodon. And then E is pointing to this green thing right here, which is your mRNA strand. So that gives you a nice insight onto what you're doing. And that is this question right here. And that's your study guide. Okay. So you may be thinking that's a lot, but again, all this is going to be turned into multiple choice questions that you will see on your exam. Give it the best shot that you can give it. The more you study, the more you have in front of you. I assume that in order to take this test and do well on it, you have to actually look at the stuff, at least have a basic understanding of it before you take the exam, and then rock and roll, get your A, call it a day. So with that, we are out, and I will see you later. Bye.